Infectious agents come in many forms, but they have basically been grouped into 5 distinct classes: viruses, bacteria, fungi, parasites, and prions. But did you know that there is a 6th modality of infectious diseases? Cancer is not normally on this list of infectious agents. Of course, some viruses, such as the well-known human papillomavirus, can cause cancers in infected hosts. However, these cancers arise independently within each affected individual as a result of oncogenic alterations in the host's own cells and remain confined to that individual. If cancer cells did travel from one individual to another, they would normally be recognized as non-self and eliminated by an intact immune system. Therefore, cancer is usually a self-limiting disease, a kind of evolutionary dead end. It either regresses or kills its host, and the death of the host marks the death of the cancer lineage. But this is not always the case. A so-called transmissible cancer originates as a conventional cancer in a single founder individual. With cancer cells then metastasizing to infect new hosts, the cancer cells themselves are the agent of transmission. Transmissible cancers are characterized by 2 defining features. First, like conventional cancers, they are clonal, meaning that all cancer cells descend from a single founder ancestral tumor cell lineage. Second, unlike human cancers, they spread through horizontal transmission, whereby viable cancer cells are transferred directly between unrelated individuals and establish new tumors in the recipient host. Transmissible cancers have previously been identified as spreading within 2 vertebrates, dogs and Tasmanian devils, and at least 10 bivalve species. Moreover, a recently published study revealed that brown bullhead catfish melanoma represents a novel transmissible cancer. Hence, this 6th infectious modality has significant effects on both vertebrate and invertebrate animals in both the terrestrial and aquatic environments. The first transmissible cancer to be identified was canine transmissible venereal tumor, CTVT, a solid tumor that spreads within populations of dogs through sexual contact. This was identified as a transmissible disease and was first experimentally transplanted from dog to dog in 1876. At first, scientists didn't know what caused the disease, but transplant experiments, followed by cellular, molecular, and genetic studies, eventually revealed the truth. CTVT spreads as a single clonal transmissible cancer lineage. CTVT is present in feral dog populations throughout the world, having been reported in at least 90 countries on every continent except Antarctica. An analysis of the CTVT genome concluded that the cells have been spreading as a single cancer lineage for 10 to 12,000 years, and likely arose from an early dog most closely related to Alaskan Malamutes. Although experimentally transplanted CTVT often undergo spontaneous regression within a few months, naturally occurring infections can persist for much longer and do not invariably regress. Thus, CTVT represents one of the oldest continuously surviving somatic cell lineages, having evolved for millennia as a clonally reproducing parasitic cell lineage that has long outlived its original canine host by more than 500 generations. The first observation of a Tasmanian devil suffering from devil facial tumor disease, DFTD, was in 1996. DFTD is a solid facial tumor that spreads from animal to animal through physical contact when devils bite each other. The disease continues to circulate within wild Tasmanian devil populations and represents a major threat to the species, although conservation programs, including insurance populations and efforts to develop potential treatments, provide hope for its long-term survival. More recently, a second, apparently completely independent, lineage of DFTD, termed DFT2, was identified in a small number of animals, suggesting that if the conditions allow, transmissible cancers may arise multiple times. Notably, the 2 lineages have different origins. The original DFTD lineage, DFT1, arose from a female devil, whereas DFT2 originated from a male. Because the first reported DFT2 cases occurred exclusively in males, it has been proposed that female devils might have some capacity to recognize and reject DFT2 cells as foreign. However, the limited number of documented cases currently prevents firm conclusions about sex-specific immune responses. Transmissible cancers are not limited to vertebrates. They have also evolved in marine bivalves, including clams, mussels, and cockles. These diseases are collectively known as bivalve transmissible neoplasias, or BTN. Like the transmissible cancers of dogs and Tasmanian devils, BTN spreads through the transfer of living cancer cells that belong to the same clonal lineage. However, unlike vertebrates, bivalves do not need direct physical contact. Instead, cancer cells are released into the surrounding seawater where they can survive long enough to infect new hosts. Scientists have identified transmissible cancer lineages in at least 10 bivalve species, and genetic evidence suggests that these cancers evolved independently on at least 10 separate occasions. While most remain within a single species, some have even crossed species boundaries and successfully established themselves in entirely different hosts. BTNs are fatal leukemia-like cancers. The phenomenon of transmissible cancer has recently been discovered in another animal group, fish. Researchers identified a transmissible cancer lineage in brown bullhead catfish from Lake Memphremagog, Canada, where affected fish developed black, melanoma-like skin lesions. Tumor cells from different fish were genetically much more similar to each other than to the healthy cells of the fish in which they were found. This indicates that the cancer itself is spreading between individuals, with the tumor cells acting as a transmissible infectious lineage. This represents the first confirmed case of transmissible cancer in a fish species, and the first known example occurring in a freshwater environment. The exact route of transmission remains unknown, but the researchers suggest that close contact between fish or transfer of cells through the aquatic environment may facilitate the spread. This discovery demonstrates that transmissible cancers are not isolated evolutionary anomalies, but a phenomenon that can emerge independently across diverse animal groups. Following examples in dogs, Tasmanian devils, and bivalves, fish now provide another striking example of a cancer cell lineage that can survive long-term, evolve independently, and spread between hosts. You may be wondering whether this phenomenon can also occur in humans. As mentioned at the beginning of the video, human cancers are also fundamentally clonal in origin, but they do not spread through horizontal transmission. This is one of the key distinctions between human cancers and naturally occurring transmissible cancers. Transmission of cancer from person to person is exceedingly rare. Under normal circumstances, any cancer cells that somehow pass from a donor to a recipient are recognized as foreign and eliminated by a functional immune system. Consequently, cancer is not considered a contagious disease in humans, and individuals with cancer do not infect others with their disease. However, human-to-human transmission of cancer has been documented in several exceptional circumstances. Documented examples include the inadvertent transfer of malignant cells through organ transplantation, and the rare passage of cancer cells from a pregnant mother to her fetus. Another extraordinary case involved an individual with advanced HIV/AIDS, who developed a malignancy originating not from human cells, but from proliferating cells of a dwarf tapeworm. Importantly, in each of these situations, transmission occurred because the normal protective barriers against foreign cells, particularly immune surveillance, were severely compromised. Reports of cancer transmission in immunocompetent individuals are exceedingly rare. The few known examples involve accidental occupational exposure, such as the implantation of tumor cells into a surgeon's hand during an operation, or a laboratory needlestick injury involving a human adenocarcinoma cell line. In both instances, the resulting localized tumors were surgically removed and no recurrence was observed. Thank you for watching the video. If you liked it, like and subscribe.